Mm. And I guess we're live. I'm sorry, it started a little bit late, but I had to shower because I was grimy, and this is me with wet hair. So I guess for the next 2 hours, we'll watch my hair dry too. Anyways, hi guys. This is me trying to solve the recent paper four live on YouTube. It is going to be a nightmare. It's going to be 2 hours. But before that, just a few disclaimers. I am not the mark scheme, okay? I didn't set this paper. I'm just predicting the mark scheme. I'm just seeing it as a student as well as a teacher. And just trying to predict what the answers might be. Yeah, so don't take my answers as the golden word, okay? It's not the real mark scheme. This is before the mark scheme will be coming out. Okay, that is a disclaimer done. I also want to start this stream by saying that I'm doing this for two groups of people. One group of people would be the people who just took paper four, and are really stressed out about how it went, and they just want a place to rant or complain, and just feel seen because it was a really hard paper. The second group of people that will be watching this will probably be people who are not taking didn't take this paper for and are taking it in the future, and you're watching this because you want to see me discuss the paper. And yeah, I guess it's for you too. So maybe you will see the thought process as we run through a paper. So yeah, honestly, I don't know how this is gonna take. It's a 2 hour paper. The last time I did a live, I learned that it takes more than an hour to discuss an hour and 15 minutes paper for sure. So I'm gonna try my best to keep this under like 2 hours though, because learning from that, I think things will be too draggy if I try to do all the details and yeah, discuss every single mark point. So I'm not gonna do that. I'm probably gonna write general directions towards the mark point and like verbally just hash it out instead of like write it down. Anyways, if you are watching the stream right now live and you have any questions about the paper, I think you can put that in the chat too and I will respond. And we have one question. Someone said, but how? The papers aren't out yet, right? Yes. And as you can see on my screen is a scanned copy of the document. I work at an exam center and the papers are locked down for like 24 hours. And then the papers are made available to not just the staff who work there. Like we can take the paper and we can look at it because by now the paper is done, it's submitted to Cambridge, everything's locked in, and there's no chance of the paper leaking anymore after 24 hours. So we are allowed to have access to paper, so I'm very privileged to have this paper, I suppose. This is the variant 3, paper 4 variant 3. And I only have this variant, okay? Don't ask me if I have other variants. I don't, because I got this from my exam center. So yeah, I don't have the other variants because I don't live in multiple countries at one time. Anyways, with all that said, disclaimers done. This is again not the real mark scheme. This is a live stream before the actual paper and mark scheme have been released. And this is just me going through the paper and telling people how hard it is. Okay, before one more thing before I start though, one more thing, just one more thing. If watching this makes you feel anxious at any point, please feel free to not watch this stream. I don't think it's actually quite. I mean, I think there are people who may find it helpful to rant online and see people complain about the paper and break down the paper, and this is how you manage your stress and anxiety. But for some of you, maybe that might not be very productive. So if you find this anxiety not productive and find this complaining not very therapeutic, please do not watch the stream. Please don't put yourself through that suffering. Okay. Now you can start. This is a 100-mark paper, by the way, and it has 10 questions. 10 questions, and it has 24 pages in total. It is very big. And again, I'm going to try and go through in general as quickly as possible. If you have any questions, let me know. Okay, so the first question is quite standard, I think. It is a cute animal. There's always a cute animal in a bio paper. I think this camel's pretty cute. Anyways, it is found in only the desert regions of Mongolia and southern China. Okay, paper. Right, thank you. And it is at risk of extinction in the wild. Okay, and there's only 950 wild camels left. Suggests why the Bactrian camel has become critically endangered. There's only 2 marks here. It's a suggest question. You can give it your best shot and talk about anything really. So my answers would be things like, okay, maybe there's habitat loss because, you know, climate change, right, would make places who are not supposed, climate change maybe of some, like the desert became warmer and not so suitable for living, something along the lines of habitat loss. It could be also due to things like human development and building of infrastructure, so like human factors because of that causes habitat loss. Bear in mind that this is in the desert, so that don't say cut down trees. There's no trees in the desert, right? Mostly no trees. So yeah, keep on those lines. What else would I write? I would probably write things like, okay, maybe it doesn't have food, right? So less food supply, whatever that is. Maybe the plant that they eat are not found in abundance anymore. Or maybe they have an increased predator. I know it's a really large animal, but I didn't know what the predators are. But maybe they are predators, right? Maybe there are human activities like overhunting. I'm not sure why people would hunt camels. Do you guys know if camels get eaten? I mean, probably for their fur. I mean, the desert is really hot, but it's really cold as well in Mongolia, in China. It can be. But yeah, something along those lines, any two will do. Moving on, this is easy. In the role of IUCN, this is again a past year, very standard question. I'll probably write something along the lines of, okay, monitor and categorize threatened and endangered species. I'm just gonna leave it at threatened species. I'm gonna write something about education if I were writing this paper. Probably something along the lines of conserving biodiversity or the species, right? Protecting the species. I would also talk about research and reporting, right? These are all things IUCN could be doing, would be doing, is currently doing. Yes. Again, still easy. Moving on. All right, this is quite new though. This question, 1C, it says that zoos use assisted reproduction techniques such as embryo transfer in their captive breeding programs. I've never seen an embryo transfer question in a very, very long time. I don't think I've seen it before. But the first thing that comes to mind is IVF, because you need to have the embryo to transfer into a mother, right? So you must have formed the embryo in the first place, and they want the procedure, right? So okay, probably talk about IVF. Again, not going to look into the details as much compared to my paper 2. Something about super ovulation, like injecting the female with a hormone to cause super ovulation, then collecting the ovum or oocytes, both are accepted probably, and sperm from the male ovum from the female, and then mixing them, and then seeing if it works, seeing if it forms an embryo, and then after that, moving the embryo into after that you do embryo transfer to maybe like a surrogate mammal. I mean surrogate mother, or say, I don't know, any mother, the host mother or the donor female which donated the ovum in the first place. Yeah, I think that's the extent that you learn. So this is what comes to mind. Tell me if you know other points in the chat. So yeah, first question, still alright. Embryo transfer, quite new, but if you have done, the study work, should be okay. Moving on. So next question, photosynthesis. Name one chloroplast pigment. Easy. There's chloroplast A, there's chloroplast B, there's xanthophyll, there's keratin. Do I need to write it down? No need lah, like you guys know, right? Chlorophyll A and B, xanthophyll. Any one will do, by the way. They said one. Xanthophyll, or what did I say? Keratin. /B. Next, very typical past year question again. They want you to differentiate between the absorption spectrum and action spectrum. It's only 2 marks, so they really just want you to say the definition. So the definition would be absorption spectrum is to figure out the wavelengths of light, right? What wavelengths, how high, sorry, let me rephrase that. How much light is absorbed by pigments at different wavelength. I'm sorry for the ugly handwriting. Trying to keep this short. That's absorption spectrum. How about action spectrum? Action spectrum is not about the absorption of light, rather the rate of photosynthesis. Also at different wavelengths of light. At different wavelengths of light. Done. Easy. Next, explain why temperature can be a limiting factor of photosynthesis. Again, very standard question in past year. Still alright, not too bad. The idea is high temperature. Sorry, let's talk about low temperature first. Low temperature, low kinetic energy, right? And therefore, enzyme rate of reaction would be pretty low, right? And photosynthesis requires enzymes, right? There's enzymes in the light dependent and the light independent. And we can talk about the opposite as well. And when it's high temperature, there's a very high kinetic energy, right? And this would cause enzymes rate to increase at first, but if it's too high, if it's very high, that is going to denature the enzyme. And therefore, again, the rate will be too low. Okay, will be very low. I think the third mark point, other than this enzyme thing, most likely will go to, because this is kind of related to AS, like enzymes, AS related. So maybe something along the lines of collisions, like low temperature, low frequency of collisions, less ESC form. There's one probability. I think there's another, because it's about photosynthesis, right? So I think it might want to hear you name a specific enzyme for a specific process. For example, Rubisco. Yes, which is involved in the Calvin cycle. That's a very important enzyme in the light independent stage. And this Rubisco will be very affected by temperature. So that's how it can be a limiting factor in photosynthesis. Oh wait, it says limiting factor, right? Scratch that. I don't think it's low temp. It's high temp, because the graph goes like this, right? There's temp, and there's, oh, wrong. Rate of photosynthesis, then there's temp, and then it goes like, wee, and then it plateaus. So it's not about low temp, my bad. It's about high temp, denaturation, and maybe a specific example from photosynthesis. It's late, okay, it's 9. I'm sorry, I'm human. Okay, but it's a quite typical question. I think if you look in past years, you will find a more accurate mark scheme. Next, outline how the light-independent stage of photosynthesis leads to the production of carbohydrates, such as starch. So basically, they want to hear the process of RUBP becoming GP, becoming TP, and then back to RUBP. TP is what triosphosphate is what is used to make things like starch or amino acid or glucose, right? Catalyzing this reaction is Rubisco, and this helps fixes carbon dioxide, and in the process this requires ATP and NADPH, also known as reduce NADP. I'm quite lazy to write it in full sentences, but basically it's this process. You describe this process in 3 marks, with the emphasis of triose becoming starch and some triose going back to regenerate RUBP. Yes, just write it in words. Next question. Not so bad so far, but next question. So 3A talks about mitochondria now. So it's not photosynthesis anymore, it's focused on respiration and how the structure of the mitochondria is related to the role. Great. And what they did was they took the outer membrane off, right, and kept the inner membrane intact. Very interesting. So they asked, right, they said that they placed the outer membranes in pure water, and this resulted in bursting the outer membrane. However, the inner mitochondrial membrane does not rupture and remains intact. So why is it intact, even though it is placed in pure water? And the only reason for that, right, suggest and explain, so it's based on your understanding, would be because the water potential is the same. Because that's what you learn in AS, right? That if it doesn't rupture, it means that the water potential inside and outside is the same, which means the water potential in the matrix, right, is the same as water potential of pure water. And it's really unusual, obviously, and this is a guess, right? Because this mitochondria doesn't have a cell wall, correct? So this is the only explanation here. And don't forget, it says suggest and explain. So why is it that there is same water potential? And the best guess in my brain is that it's because the ions are always actively pumped, right? Hydrogen ions especially are actively pumped into the mitochondrial membrane. So pump across the inner membrane, but into the intermembrane space. Right, so much so, right, that inside is pretty much almost all water. All the ions are pumped out into the intermembrane space. So that's why the outer membranes will remove and there is rupture at the intermembrane space, but inside it remains intact. Tell me if you have any other alternative answers, but that's what I'm thinking of. Next question, name 3 molecules other than coenzymes, okay? Other than coenzymes. I thought really hard about this one. That are found in the mitochondrial matrix and explain their role. So it's 3 marks, so they want you to name and explain their role. So again, this is respiration, so I'm thinking about the link reaction, because glycolysis happens in the cytoplasm, we're thinking about the mitochondrial matrix, right? So the link reaction must be, it happens in the matrix. So what do we need for the link reaction? We need pyruvate, which is a product of glycolysis and used in the link reaction, right? And then is converted to acetyl coenzyme A, but acetyl coenzyme A is a coenzyme, so probably not great. I'm not sure if they will accept acetyl coenzyme A as carrying 2 carbon to the next stage. I feel like it might be a coenzyme, so I don't think it's a safe choice at all. So that is what I'm thinking of. And then what happens after links is Krebs cycle, right? It's GLKO. Girls like Korean oppa, right? So there's glycolysis, not involved here. Link reaction done. Krebs cycle. Krebs cycle, we start with the oxaloacetate. If you refer to my other videos, I have a ridiculous story to help you remember Krebs cycle. And this converts to citrate, and these are the only 2 molecules you actually learn within the Krebs cycle. Oxaloacetate is 4 carbon and citrate is 6 carbon, right? And these two talk about how they're used in Krebs, right? So oxaloacetate combines with acetyl group and it makes citrate, and this is the first part kind of the Krebs cycle. Now there are other things as well. I think these are weaker answers because they're not as prominent. What I was saying, carbon dioxide, because yes, they don't really have a role per se, but they are waste gas. Does that count? So I'm not sure if this counts. The other one that I can think of is ADP and inorganic phosphate, because you need that to form ATP, right? So that could be an answer as well. Again, I'm writing brief notes. You write them in full sentences. I hope you did. This is not by no means a mark scheme. I'm going to say the disclaimer like every now and then. Next question. It says inner membrane contains a very high proportion of this molecule called cardiolipin. This is not a molecule that we have learned, but they tell us that cardiolipin makes the membrane impermeable to some ions. And the reason why it is impermeable, sorry, my English. They want you to suggest why it has a high proportion of cardiolipin that makes it impermeable to some ions. This is a very roundabout way to ask you, why does the inner membrane need to be impermeable to some ions? And the answer is quite standard throughout the past years, which is to maintain electrochemical gradient across the inner membrane, right? So we want to pump things out, but we don't want the ions to move back in to the matrix. We want to maintain the electrochemical gradient. There's a high concentration of H plus ions in the intermembrane space, much higher compared to the matrix. So that's why. Next question. Still okay so far, right? Next question. In further experiments, it's found that in an intact mitochondria, they tell you that there's a membrane potential, okay, means there is a charge difference, and that this transport of ATP, ADP, and inorganic phosphate shown here, again, this is not something we have learned, this is something they are giving us new information of, right? So this fella and this fella, right, is actually driven by this membrane potential across the inner membrane that we talked about just now. First question. Explain how hydrogen atoms from reduce NAD and FAD, right, lead to a membrane potential forming. So basically they want to hear what happens to these hydrogen atoms. And so they really want to hear about the process of oxidative phosphorylation. It's not purple, make it purple. Oxidative phosphorylation. So, yep. How does that work? So hydrogen atoms consist of the ion and the electron. So we can talk about it splitting and then how the electron moves down the electron transport chain, and this releases energy, and this energy is used to pump hydrogen ions into the intermembrane space. I said intermembrane space a lot in this video, didn't I? And yeah, this causes that difference in membrane potential. Did I write anything else? No, that's pretty much it. That happens for both reduced NAD and reduced FAD. They didn't really ask about the formation of ATP, so I think the mark scheme kind of stops here and doesn't really it doesn't really talk further into ATP, the usage of the proton gradient. The usage of the proton gradient in the formation of ATP synthesis. It just stops at proton gradient because that's what the question is. Next, suggest and explain how inorganic phosphate is transported across the inner membrane of the mitochondrion into the matrix. Okay, so this is based on the picture, I assume. So we can see here that hydrogen ions are moving from the intermembrane space, which is high in hydrogen ions, by the way, right? And this at the same time, right, inorganic phosphate is being moved into the matrix as well. And this reminds us about AS concept, also in A2, about co-transporters. This is a co-transporter for sure, right? And even the question tells you that they rely on the membrane potential in order for ATP, ADP, and inorganic phosphate transport. So co-transporter is the answer here, right? So you probably need to explain this a little bit and say how hydrogen ions are really traveling down the concentration gradient, whereas the inorganic phosphate might be transported against the concentration gradient. And this transport is actually a form of secondary active transport because it utilizes the gradient of hydrogen ions in order to transport inorganic phosphate. Yeah. Next question. Suggest the advantages of linking ATP transport to ADP transport across the inner membrane of the mitochondrion. Okay. So they are talking about this area here, again, new to us, and you just gotta look at the question and really think it through. So this is me trying to figure out what they want. So it seems that one ATP goes out and then the ADP can go in, which is good, right? And in my brief notes for myself, I wrote, haha, supply equals the demand, right? What you're using as a substrate is being transported inside, and the products are being transported out. And I guess that is the advantage, right? So the substrate, right, which is the ADP, is going in, the product is being moved out. So, and this is at the same time, right? So there will always be substrate going in and there will always be product going out. And that's good, right? So there will never be a lack of substrate and the product will always be transported out. Okay, and the best part is because ATP kind of breaks down into ADP and inorganic phosphate for when it's used in the cell, and this is transported back in so that this can be recycled. So I think there is one mark that goes to really talking about how ATP is used for other processes in the cell, resulting in ADP, which is then transported into the mitochondria and used in the ATP synthesis process all over again. Does that make sense? I'm thinking, like, is there any other answers that can fit here? If you have any other ideas, feel free to comment it or write it in the chat as well. My brain is going a blank now, so I'm moving on to question number 4. Question number 4 is about ADA. Again, this is not very common. I haven't seen an ADA skit question in a very long time, but it seems okay because the first question says, outline the principles of genetic engineering. And we can do that. I think they are really talking about recombinant DNA technology here. So taking a little bit about recombinant DNA technology. So taking a dominant normal allele from like someone and using my brain just stopped working, and making more of it, maybe using recombinant DNA technology or using PCR, and then putting it into a vector, and the vector could be, a retrovirus, a liposome, or just simply a gene gun, and using that vector to deliver it into the body so that this normal dominant allele can be expressed, right, in the human host, right, and therefore cure the disease. But the disease has to be caused by, the assumption is that the disease must be caused by a defective recessive allele. Yep. There's notes in my video, so I'm not going to write this in full again, but I think this is the way to go for this question. So far, I feel like the paper is okay. Really, so far it's okay. But by the way, if you just tune into the stream or you're watching this at a later date, again, disclaimer, this is not the mark scheme. This is me guessing the mark scheme. This is literally a few days after this paper, so I don't know the real one. I'm just guessing, like, in what general direction it's going in. Okay, next question is about gene therapy and using the gene-corrected stem cells, right? Using gene-corrected stem cells and infusing them, and one is enough, okay? So the context is actually for gene therapy, it usually takes like multiple tries in order for, because you usually use a vector by some sort, and this vector will most likely brain going blank. True to exam conditions. What did I want to say? I really forgot. Oh yeah, now I remember. So the vector literally like delivers the DNA, and the vector can be attacked by the immune system because the vector might be recognized as non-self. So it activates the immune system. So it will degrade over time, usually. And whatever RNA or DNA that's inserted, that is going to degrade over time as well, especially if it's not inserted properly. So it's quite rare to have a single infusion that is enough. So they're asking you why this time a single infusion is enough. Why not multiple times? And the answer lies in the question, right? It's because of stem cells. This is again an AS related question based on your AS knowledge. And the idea is stem cells can self-renew. They can make one cell that, when it divides by mitosis, right? One cell can differentiate, and the other cell is one of itself. So one cell of itself, one of itself. So this is called cell renewal. And therefore, since it's a stem cell, it wouldn't degrade over time. It will self-renew, and therefore a single infusion is enough. I probably think more marks would be allocated for mitosis, and talking about that they are genetically identical and they would all contain the proper correct gene. I think that was quite, like, you have to understand the context of where it's asking in order to answer the question. But still alright, okay, it's still okay. So phew, not too bad so far. It gets crazier towards the end. Explain why a promoter has to be transferred as well as a desired gene. Again, this is quite a standard past year paper. I've seen this a couple of times. And the idea is that RNA polymerase will need somewhere to bind. And if RNA polymerase can bind, it can, or transcription factors can bind, then transcription will occur, and transcription occurs, and therefore the gene will be expressed in and made into protein. So promoter has to be transferred for gene expression into protein. Next, a modified retrovirus is used to insert the gene into DNA of blood stem cells. Suggests two ethical considerations of retroviruses. So here we can talk a lot about it. Again, this is a typical past year question. So talk about, say, how retrovirus may have the potential to cause diseases. So you need to remove that first. You need to probably talk about how it can multiply inside the host. There may cause disease. It also may cause allergic reactions because it's, after all, a virus, and it may be detected as non-self by the immune system. You can also talk about how when retrovirus incorporate the DNA into, right? So retrovirus is RNA virus, right? So it takes RNA virus and it converts it using reverse transcriptase to cDNA, and it will randomly insert the cDNA into, it could, right, randomly disrupt genes, right? It could insert into a gene, and that would be a problem as well. So you can actually specifically engineer this retrovirus in order to not have all these problems. But since they ask, this is what we consider when we talk about retrovirus. Next question. C. Gene therapy. Why is it... What are they asking? Oh, they're asking why is it more potential cure for Huntington disease? Why gene editing for Huntington's and not gene therapy? And for this, you need to be familiar with Huntington disease. Realize that it's caused by a normal dominant allele. Faulty dominant allele. And this only kicks in at a later stage in life. It also not in a very accessible part. It usually affects the brain and actually the whole body. So it's very difficult to cure, right? Using gene therapy, using normal methods. You probably want to use gene editing to edit out, or like turn off the faulty dominant gene so that this is not a problem anymore. Yes. There are 3 marks, I know, and I said a lot of things in between, so somewhere there is the direction, I think. Okay, so I'm gonna move on. Again, this is like a really quick like walkthrough, not like a specific mark scheme. I tried doing that for paper 2. It was like really long. I'm just trying to keep this short. Again, if you're just trying to stream, this is not the mark scheme. This is me trying to predict the mark scheme. This is me looking at the paper of just a few days after paper 4. Okay, now we are at question 5. This question was the killer. It literally was. I stared at this question for so long. I let other lecturers look at it, and so many different teachers gathered around the paper for so long. I couldn't figure out this question. Let me break down the most likely conclusions we came to. This was the killer. I'm just gonna highlight it just to make a mark. Anyways, they told you that there is this alpine plant species. They're not considered trees, okay? They're alpine plants, and they are defined by plants that live above the tree line, which is where trees can grow. So imagine like a little like mountain. There is like trees here, and then there's trees here, but at one point maybe the mountain's too high, right? And trees cannot grow as much anymore. But these alpine plants can grow, right? Let me draw that in a different color. These alpine plants can grow like on the mountain top, above where the tree line is. So here, tree, here, it's a tree. I know, ugly, but it is a tree. But alpine plant species are only these green fellas above the tree line, and they tell you that currently it is at 1,200 meters, and this will become important later on, I think. Okay, the first question is interesting though. The alpine areas nearest the South Island of New Zealand. New Zealand has 2 islands, by the way, one north, one south. So the nearest alpine areas to South Island is like 1,500 to 2,000 km away from the sea. How did a large number of alpine tree species, not tree, develop on South Island? Because many of South Island alpine species live nowhere else in the world. So I thought to myself, like, is it sympatric speciation? Is it allopatric speciation? Then I thought to myself, hey, it's an island. It doesn't have it has nowhere else in the world. So they're not giving us a clue here about like what species it branched off from, and like it's a population that's split into half or anything like that. It seemed to have independently developed. So I feel like I have a feeling that this question is actually about natural selection, right? And how they became better adapted to grow above the tree line, and eventually they became their own species because they were so different from the normal trees. So I assume that they actually came from the normal trees, not from elsewhere in the world, because they're so isolated, right? And the way this question is structured is strange. So natural selection, I always have like a cheat code up, and that is talking about variation first, talking about there's different types of plants, and talking about adaptation. So the fellas that have best adapted to that particular, like height would survive. And there's always an idea of survival and reproduction. So, and then there's the idea of over time, the allele frequencies of these beneficial trait will increase over time. And so I think it starts from there. And then the idea is that it becomes so different because it is, so it's kind of like sympatric speciation in a sense, I suppose. It becomes different from other plants and becomes its own species. Maybe. This is my guess. I don't think it's straightforward allopatric or sympatric, but feel free to disagree with me. Before I move on, I saw a question. Actually, I saw it a while ago, but I didn't know when to bring it up. Clee and Herdal says, what do you think the threshold will be, Miss Bay? I have no idea. I think it'll be quite low because it's quite an unusual paper. Like there are questions which so far you have seen, like that I said, we have seen this in past year before. It's okay. Not too bad. But there are questions like this, which is very unusual, and I don't know what it wants. So these are places where people might stumble. Let's look at the next question, which was my headache. By the way, there is like a tiny line here that reaches 1200, which is not reflected in in the scanning process because I use my phone. But yeah, the current tree line is 1200, and the current mean height of the mountains is 2400 meters. Okay, so they show you two graphs, right? One is a dashed line, so this is the mean mountain height. I'm just going to draw mean mountain height. And then there is also height of tree line. So all this zigzag solid line model is about the tree line, and they tell you, just FYI, that the tree line is higher when the climate is warmer. So most likely in this peaks is warm, and then when the climate is colder, here, right, it's probably the tree line is lower, and that's probably why. And that makes sense because if it's warmer, more trees can grow, therefore the tree line can be higher. If it's colder, then even the flat surface, no mountain place would be cold, and therefore a tree line will be very low. How about a tree line that dips below zero? This might be trees, it could be flooded, or it could be in a basin, right, which is not flooded, like that is under sea level. So okay, we'll just accept it. The question they ask is very interesting. Question they ask, it says, identify with reasons the time period when South Island's alpine plant species developed. So they want to know at which point did the alpine plant species had the opportunity to become its own thing. So I'm thinking to myself, okay, they told us that this is the current data, but that tells us nothing about the time period. I can extrapolate, but that doesn't make sense either, because the alpine plant species is always going to be, above the tree line, and the tree line changes, right? So any trees above the tree line could be alpine plant species. So after many discussion with my colleagues, which I dearly love, just saying, shout out to you guys. I don't think they're watching, but still, I love them a lot. We think that it is not extrapolation like this, but it is at the point which the mountains surpass all the tree line, which means there are plenty of mountains, so plenty of space, in order for the alpine plant species to develop. So we think an estimate would be, if you draw a line down here, it would be around like 1 million years before present. So it developed 1 million years ago, up to the present time. So most likely giving a range would help. So maybe 1 million years ago till now. 0 to 1 million years ago. So this region. And why? Because, well, the mountains were taller than the tree line. Yes, that's it. I stared very long and hard this question. I can't think of any more straightforward, normal answer. I think if you can extrapolate it, you'll end up like somewhere at this point here, and you move it down, it'll be around 1.6 million years ago. And I feel like, okay, that would be true if you fix the tree line. It's 3 marks, right? So no harm writing, I suppose. I'll probably write something, just a backup point, going like, oh, it probably is at the point where the mountains are above 1,200 meters, at least, right? Because this is where, again, the alpine plant species has the space to grow. Space to grow. And time to develop. So yeah, that's generally what we think. I'm not sure if it's right or not. Just guessing. We will know when the real mark scheme comes out. Next, suggest how DNA sequence data could be used to confirm the time period you identified in 1. So they're asking us, how do you use DNA sequence data to figure this out? A time period, and I was thinking to myself, maybe it's just general, like, okay, you take the DNA sequence data of all the alpine plant species, and then you basically compare the DNA sequence and measure relatedness, genetic relatedness, and if you do this enough, you will actually figure out which plant, because alpine plant species, I assume that there are many types, and therefore you can DNA sequence a lot, and then you can confirm the time period you identified. So you find like a map, and maybe you get like you also like DNA sequence the trees around it just in case, and see how it maps out. You can see and approximate like how long it has been. So I think that's the answer. Do you have any other thoughts? Let me just flip through my notes. Yeah, looks fine. It looks fine. Next, question number 6. How vigorous uniform varieties of maize were developed by selected breeding? So, how does vigorous uniform varieties were developed not by like normal things, not by like genetic technology, but by selective breeding? Hold on a second. I need to go do something. I'm doing a live stream, so you talk over there. Sorry, background noise issues. Okay, before I move on, let me answer a question. Meme X said, "Ma'am, can I do paper 1? Paper 1 hasn't happened yet, right? Did it happen already? I heard it hasn't happened. If it hasn't happened, I don't have access to the papers, guys. Yeah, I'm not a paper leaker. This is a paper that's already been set for. Just saying. Here I am, just guessing the mark scheme, not knowing the mark scheme, perhaps. Anyways, this 6A, it says, how do you make uniform varieties of maize by selective breeding? And this is actually part of the syllabus. Basically, you do a pure breed, like one group of pure breed and a second group of pure breed. So you breed it, and it might be inbreeding repression. But then, last but not least, you cross it, right, at the last step to get F1 hybrid plants. And these hybrid plants will have hybrid vigor, and if you plant them, they'll all be the same size. Ta-da! And this is part of the syllabus. I have it in my notes as well. I talk about it under selective breeding, one of the examples that are in the syllabus. Again, I think this is possible to do if you have studied the content, but it's rarely seen in past years. So if you're just basing your knowledge on just spamming past years, you might have trouble with this question. It's quite rare. Next, IR8. They said it developed in the 1960s, and it was developed by bringing together 2 varieties of rice. This one has high yield, and this is dwarf. And they're asking you, why is dwarf a good thing? Right? Why is it an improvement of Peter? and it's most likely because, right, the edible grains part looks like is reduced height means it is less affected by wind. It's tall, the center of gravity is high, therefore if the wind blows it will fall, right? But if it's short and dwarf and still has the same yield, it is better in terms of wind resistance. Yep, and this would be more what's the word? Be a more viable plant, not destroyed by storms or anything like that. Cause rice, it's kind of a tropical thing, so we have storms. In 20 Next question, they said that they sequence IR64 sub 1, which is actually breeding together of two varieties, FR13A and IR64. And IR13A has low yield but flood tolerance, and this one has high yield. So most likely is to identify the gene, right? If you sequence all these three data and you look at how it's different, you can probably identify genes for flood tolerance and genes for high yield. Basically genes or alleles that contribute towards this, and therefore will be great for maybe genetic engineering of future plants. But here we are still talking about selective breeding. After few generations of breeding, the scientists cross this with IR64, which is the high yield plant. Why did they do this? Again, this has to do with something you learn in your syllabus, right? The idea that there is inbreeding depression. If you inbreed a plant too much. I just wrote in depression. Inbreeding depression. And therefore, crossing it out would actually result in hybrid vigor, which allows the plants to be better overall. So yeah. Next question. Part C. This is quite interesting, right? They have a wild type, a dwarf, and a dwarf variety. You can see that the wild type is normal, and then after gibberellation treatment, it is very high. Gibberellin is a plant hormone that affects growth, right? This is a dwarf, and it's not growing, but after gibberellin, it has increased growth. This one, in both treatments, there is no effect. So why? Why is it so? Right, so this has to do with the allele, right? For gibberellin, we usually use the LE to figure it out. So this is big L, dominant. This is codes for active gibberellin, and this is inactive. So my guess is for A, it has the recessive allele, and therefore just small l, small e, right? And therefore inactive form of gibberellin. And after you give it treatment, it can increase because it's just inactive, but everything else works. So as long as you give it the correct gibberellin, which is active, then it will grow. It will have a lot of growth. However, B, there is no problem with this. So it has nothing to do with the active or inactive gibberellin, I think. Because it's a hormone question, and hormones always have receptor, B doesn't respond to treatment because it has a faulty receptor. So even though you put like high amounts of gibberellin, it doesn't result in increased growth. And you can explain this a little bit, and that should score you 4 marks. Ta-da! I think this was quite hard. I'm gonna give like a yellow thing here. It requires you to really understand hormones, link the concepts in your brain. It really tests understanding, not just blind memory. Next, this is a transcription factor in A. thaliana, which is the plant. So this is a eukaryotic transcription factor, and they're just saying, outline the features of this transcription factor. So how does it help transcription, basically? So it can bind to promoter, right? It can recruit like RNA polymerase, and therefore like increase transcription rate, right? And all of those things. This is eukaryotic, so this is not limited to the idea of leg operon, so it could be anything, and it's 2 marks, so there's a lot to write here. But this is actually a quite rare question, I would say. Like, I've not seen a question on transcription factors on eukaryotes until now. Plants are eukaryotes. It's always been leg operon, not really eukaryotes. But here it is. Now we have it. I'll update my notes, I think, when the mark scheme comes out, to include all these different things. Alright, next question. This is question 7a. They tell us that this gene has 2 alleles. One codes for black fur, the other one ginger fur. And these black fur and ginger fur alleles are co-dominant, and they are on the X chromosome. So yeah, show how the cross between a tortoiseshell female and a ginger male will work out. So tortoiseshell female, right, we start with XX, right? It is on the X chromosome. It's co-dominant. So we have small b and small g. Because it's co-dominant, right? So don't use big B and small b, right? It's the same gene, but co-dominant. There's no dominant and recessive. It's both the same, both expressed at the same time. The ginger male, however, because it's an X-linked gene, it's on the X chromosome, so this is the only possible genotype. The gametes would be this. And since they didn't give us space to draw a table, I guess they don't want a Punnett square. Just be very careful to make sure you include all 4 different genotypes. So XB, XG, XB, Y, and then XG, XG, and XG, Y. So what's going on here? Right, what's the phenotype? Tortoiseshell female. This is a black male. This is a ginger female. And this is a ginger male. Ta-da! And that will be 5 marks. Next, thyroxine gene. Haha. So TRY gene actually codes for thyroxinase enzyme. Thyroxinase. Again, this is one of those questions that haven't been out in a while, but this is not too bad. Like, I've seen this before once or twice. But yeah, it actually codes for thyroxinase enzyme, and this converts thyroxine into dopa, and then into dopaquinone, and into melanin. And it actually acts here and here. I'm not sure if they require the word dopa and dopaquinone in order to get the mark, but I would write all in just in case. But even if you don't write all, I think you might still get the mark. Yeah. Yes. Next. Question number 8. We are so close to the end, right? Question 8a. Right, so we have the distal convoluted tubule in A, B is the cortex, and C is the loop of Hindley. This is the easiest question in the paper. It's based on the kidney structure. You should be familiar. Next question is also a kind of common thing, but just read the question and you realize that, oh, as blood ADH increases, what they're showing us is that urine concentration, which is this solid line, increases in concentration and it decreases in flow rate. And they tell you that flow rate is the rate of production of urine by the kidney. So basically, more ADH, more concentrated, less volume. That's pretty much it. So describe the relationship shown in figure 8.2. That's literally what I said. Just write it like do this, move it here. Dun, 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 dun. And this is general trend. After you describe that, insert a data code to support your data, and you should get full 3 marks. Easy. They just want you to describe. They didn't want you to explain. So it's fine. Only the second question will ask, describe and explain action of ADH on cells of collecting duct. Right? So only now they ask you to describe and explain. And here, you can talk about it in many, like, just talk about it, right? ADH is a very common topic, common past year question. So what does ADH do? It binds to the receptor, and this receptor is complementary in shape. Is 5 marks, so you might want to include as much detail as possible. And this receptor is on the collecting duct, and this allows vesicles containing aquaporin to move and fuse with the membrane of the collecting duct, right? And this is a kind of like exocyte. I won't say, no, it's not exocytosis, because aquaporin is at the membrane, right? So this increases permeability of that collecting duct to water, and therefore there's a higher rate of water reabsorption, and therefore when the water potential blood decreases, this would increase the water potential of the blood again. I'm not sure if they want us to talk about the pituitary gland, because they just said explain the effect, describe and explain the action of ADH. They didn't say describe it from beginning, did they? So I think this is the area of the mark scheme. Second last question. Let's go. 9A is about neurons. So this is action potential in the neuron. There is the depolarization, repolarization, hyperpolarization, and refractory period. Very interesting. Next, it says action potential in the striated muscle fiber. Now this is a graph I've not really seen that much before. I realized that the time on the x-axis is like shorter. And realize that the y-axis is the same, pretty much the same, except hyperpolarization, right? So the first question is that you have to describe the differences, and this is just describe what you see. You don't need to explain, so you just say that, you know, when does it peak, right? So you can describe the peaks. This is at 4 milliseconds, whereas this is at 2 milliseconds, right? And it's at positive 40. There is this dip in hyperpolarization. You know, this fella doesn't have that dip, right? There is, it seems like from this point to the end, it only takes 4 milliseconds, right? But here to the end only takes, okay, you can just consider peaks instead. So for resting potential, to get back to resting potential, right, it's how long, and this to this is how long, right? And you realize that here is 4 milliseconds and here is 2.7 milliseconds. Amplitude wise is pretty much the same though. except that resting potential seems to be, so-called resting potential, is lower at the striated muscle fiber than here. The peak is the same, but the resting potential is not. This one here is -70, and here is -90 millivolts, and that can be compared as well. They say they want differences only, so there is no similarities needed. I'm lazy to write it down, but basically I did that verbally. If you manage to describe those things in full sentences, you're good to go. Next question. Next question tells you that there are 3 phases in the contraction of striated muscle. This is not in the syllabus, right? This is new information. They want you to analyze the information given. So this is the latent phase, right? Which is like a little bit after stimulation. There's contraction and there's relaxation. So what's going on? What is happening in the latent phase, basically? So why is it stimulated but a muscle doesn't immediately contract? So what happens from depolarization, right, all the way to contraction? And this has to do with spreading of depolarization across the membrane, which is called what already? Sarcoplasmic, sarcomere. What is the membrane called? Membrane. Right, the membrane of the muscle cell, right? It spreads across the membrane, it spreads across the sarcoplasmic reticulum, which, T tubules first. T tubules, and then the sarcoplasmic reticulum, which is the specialized ER of the muscle. And then after this depolarization spread, what happens is calcium ions are pumped out or released actually from the sarcoplasmic reticulum, and these calcium ions bind to troponin, which then moves tropomyosin, and then therefore the actin and myosin can bind after that, because the myosin binding site is exposed. So cross-links, cross-bridge can form and contraction can continue. So it takes some steps for it to get from stimulation to contraction. It doesn't go immediately. All these things must happen first, and therefore there is a time lag. My student did feedback to me that he struggled with this question, so I'm going to mark this down as difficult. Next question. Suggest why the relaxation phase shows a gradual decrease in muscle tension. So contraction will require actin and myosin the myosin to pull actin inwards, right? And relaxation means that sarcomere would lengthen again. So you contract and then you relax. So what kind of, what kind of thing happens, right? And they want one mark. Why is it gradual, right? Why is it not quick? And my guess, again, this is a guess, is that it requires a few more steps, right? ATP is needed to be hydrolyzed in order for myosin to detach from actin. And this takes time. And this occurs without stimulation, so it takes a time, maybe. I have no idea. Honestly, no idea what they want here. I mean, it makes sense, but I don't know why. I don't know how to explain how it makes sense that relaxation takes longer time than contraction. Again, if you have any ideas, write them in the comments. I'll be happy to hear what are your guesses and answers for this question. I'm at my limits here. I'll mark it down as hard, so that's a little tick. And finally, we're at question number 10. Question number 10 is fill in the blanks. Thank God. Okay, so here it's just about homeostasis. I'm guessing that the process, homeostasis is the process of keeping the internal environment of the body in optimal conditions. It can't be external. That is part of the definition. And they are all kept at the optimum value or set points. They even give you kind of examples. When a condition deviates from a set point, a corrective mechanism is triggered, and this increase would become decrease, and this is called negative feedback for sure. They said most appropriate scientific terms, so I assume that one line doesn't means one word. So let's read the next one, which is the one I struggled with a little bit. The pancreas involved in control glucose concentration. Glucose binds to the what of cell surface membrane of pancreatic cells. So I would say receptors. That just makes sense, right? And this is how the pancreatic cells figure out whether glucose is high or there's low concentration of glucose. And these are what cells? These pancreatic cells. What are these cells which secrete hormones such as insulin and glucagon? So I'm like conflicted here. I don't think it's pancreatic cells again. So I can say alpha and beta cells. I can say islet of Langerhans cells. I think alpha and beta is the most specific though, so I'll just leave it as is. Yep. For example, the action of one hormone stimulates the uptake of glucose by respiration, and the other breaks down glycogen to glucose in the liver. And with that, we are done with paper 4. Finally. So, it's 100-mark papers. Let's go back, all right? Let's go back and look at what questions were challenging. Fill in the blanks, fine. This question, hard. So 3 plus 1, that's 4. Okay, 4 so far. 4. This is fine. Moving on. This is fine. This is fine. They're okay here. This is fine, right? I think this question is fine as well. Thyroxine, if you know it, you know it. If you don't, you don't. However, I'm just going to mark it down because I don't think people focus on specific diseases and specific things a lot. So I'm going to rate it as slightly hard. So that's 4 plus 3, which is 7. So 7, 7, 7. Next. Features of transcription factor, I feel like you can wing it. You can talk about enhancers. You can do it. I think this question is quite hard. I'll put 4 marks there. So 7 plus 4 is 11. So 11 so far. Scroll all these things. I think 6A is quite difficult because it's very rare, and people usually overlook this example. They're like, oh, so just in the examples. So 15 marks are hard to get. I feel like this entire alpine question is insane, and that total mark is 10, so that brings our total to 25 hard questions. 25 marks of hard questions. Huntington disease, I think, is fine-ish. This question is fine-ish. It's doable. You might lose 1 or 2 marks. It's not perfect, but it's doable. You know, by the way, I'm re-looking at this 4A genetic engineering question, and I'm thinking, I think I entered gene therapy. I think if it's genetic engineering, I would talk about like inserting genes, and it could be in any organisms, and this could be from another organism or another species, and it would be expressed in the correct species. So scratch my answer. I regret it. I take it back. All right, talk about how we can modify and insert and delete genes. That's genetic engineering. And we can take this gene from a different plant and express it in a different organism, which is fun. Okay, 25 marks. This question is fine if you're in the right direction. I wasn't. I don't think I'm right in this question. 25 marks so far. Scrolling, scrolling, scrolling. I think this, as long as you know what the question's asking, and as long as you can guess what they're talking about, I think you'll be fine here. I think this question about three molecules other than coenzymes might be quite difficult for some people. So that's 28. Let's add this to there just because, again, your brain needs to understand and think a lot of steps. So I'll make that 30 marks. 30 marks. 30 marks of hard questions. This is rare question. IVF, never seen it before. That makes it 34 marks. So out of 100-mark paper, okay, I hope it is comfort to you that 34 marks is considered moderate to hard questions. Like they are either rare or that you just need to answer that you need to understand the concepts and different layers of what is going on. So 34 marks is moderate hard. That leaves us with 66 easy-ish questions. I feel it's a doable paper in that sense. Like the ratio is correct. It's just that I think while doing the paper, and when I first saw the paper as well, you're so overwhelmed by the crazy hard questions, especially that alpine tree one, which I stared at for ages. This one, right? It's just so easy to get overwhelmed by these difficult questions that you forget that there are questions in the paper that were doable. And I hope that, yeah, that this question is insane. Like, I know I glossed over it, but the process of getting to this conclusion was insane. Like, a lot of people gave different answers, and these are all teachers, by the way, and all from different subject majors. So, Cambridge paper setters, if you're watching this video, can you can you can you make the questions, like especially this kind of data analysis questions, like make it have more context? Like this is really vague. It was really confusing to read it. It was very disorienting thing. I don't think it's a very good way to estimate time period as well based on heights of mountains and heights of tree lines. It's just a very weird question, okay? So don't do that to us again, Cambridge. You gave us a heart attack. But yeah, other than this question, again, everything else was okay. I hope this is a comfort to you if you really struggle for this. I have no doubt that the threshold is going to be low still because of the high percentage of moderate to hard questions. Yeah. So don't worry about it, you did your best and just move on. And if you haven't taken paper 4 yet, be warned, okay? Don't take bio for granted. Bio gets harder every year. Literally, every time I open a paper, I'm shocked by what kind of crazy scenarios they can come up with to test our understanding. In that sense, it's a very good exam because it really truly tests our understanding and data analytical skills. But yeah, don't take it for granted. Don't memorize without understanding because it really comes back to bite you in the butt. Yes. So yeah, that's it, I suppose. All the best with your exams or your life if you are moving on from A levels. Should I say like should I wish you, no, goodbye doesn't, should I make you comment, like, and subscribe and all those stuff? I'm too lazy to say that. But like if you found me useful, my notes useful, and you found like my notes and videos useful, as you can see, I'm not very comfortable with self-promotion, but if you do find it useful, feel free to share it. It is online, it's free. Only if you want a digital copy of my notes, it is available on biology.selfie.store, and you can purchase a copy there. Yes, that's it for self-promotion. I'll see you guys in other videos, I guess. Take care. Bye-bye. Oh, I have a few more questions. So no bye-bye yet, I suppose. Ma'am, can you do P1 too? Oh, I said I don't have paper 1. How should I revise for paper 1? Do my past years. After a while, all the all the common tricks are the same. Miss Bay, can you please look at paper 22? I don't know if I have it. I'll go find out if I do. I'm not sure if we did the paper 22 variant or the paper 23 in my center. But generally the paper 2 was quite okay, so I didn't feel a need to come online and complain about how hard it is or comfort people about how hard it is. But yeah, let me know if you have any questions, if there's any more, and I'll see you in the next video. Haha, bye bye.